I want to thank the organizers for inviting me to give this talk today. So today I'll be talking about engaging endogenous immunity with CAR T cells. I'm Sarwish Rafiq. I'm an assistant professor of hematology and medical oncology in the Emory University School of Medicine. My lab works on engineering novel approaches to better improve CAR T cell efficacy. And just as a uh, review, T cell receptors are the model for what chimeric antigen receptors are. And so for CARs, what we've done is taken key aspects of T cell receptors and integrated them into a single molecule in order to activate um, T cells and to recognize antigen of interest and kill them. And so instead of a TCR alpha beta chain, we usually have binding domains derived from either antibody scFvs or other binding moieties. And then we have a hinged transmembrane and activation co-stimulatory domains. And part of my talk today will also talk about armored CARs, which are CAR T cells that are additionally co-modified to deliver agents such as cytokines, ligands, peptides, or enzymes to the microenvironment in order to overcome immunosuppressive kind of microenvironments. So while CAR T cells have been efficacious in some hematological malignancies, they have been largely ineffective in most solid tumors. So this is a review of trials done with CAR T cells over the past decade. And as you can see outlined here, most of the responses are stable disease at best, or partial response at best. Sorry, and so there's a lot that can be done to improve how CAR T cells function in the tumor microenvironment of solid tumors, and to improve their efficacy against these malignancies. So we believe that CAR T cells have great promise in combination with immune checkpoint inhibitors, which are antibodies that generally block the binding of inhibitory receptors on T cells to inhibitory ligands on the tumor cells or in the tumor microenvironment. And so as a combination therapy, we believe that CAR T cells could be greatly improved when used in combination with an anti-PD-1 or PD-L1 antibody, wherein this would prevent the breaking of CAR T cell activation in the tumor microenvironment. Furthermore, we hypothesize that this combination could also reinvigorate tumor-specific T cells that may already be in the tumor microenvironment, and the killing of the tumor may lead to epitope spreading and T cell priming, leading to not only the effects that you would see with CAR T cells, but also engagement of the endogenous immune system. However, combination therapy with CAR T cells and ICI, although it is an active area of research in the clinic, it hasn't really seen great clinical efficacy. And so in this schematic, this is showing some of the combination therapy approaches used in different types of malignancies. And most of these trials also have not are either ongoing or have not demonstrated improved efficacy in combination. We think that especially for cancers that are refractory to current immune checkpoint pathways, that novel immune checkpoint pathways are needed to work in combination with CAR T cells to improve their efficacy against solid tumors. Vasoactive intestinal peptide is expressed by many different tumor types, and we believe that VIP is a novel immune checkpoint that can be targeted for the use in combination with CAR T cells. So as you can see in this slide on the top, VIP is overexpressed by different hematological malignancies such as CLL or B-cell lymphoma, as well as solid tumor malignancies such as ovarian cancer and pancreatic cancer. Our collaborators, Ned Waller at Emory University, has used engineered peptides to target VIP and control tumor burden. So, as seen in the top of these slides, these are three different tumor models: pancreatic cancer, colorectal cancer, and acute myeloid leukemia. And we can see that VIP is overexpressed in all three of these malignancies. If we use a antagonistic peptide to the VIP receptor, we can see that this can help control tumor burden in all three of these uh, malignancy types. So next, we wanted to see whether VIP and the VIP-VIPR axis was a novel immune checkpoint for CAR T cells in particular. CAR T cells have distinct biology, as I mentioned in my previous slide, from T cells. And so it's important to know whether this axis is playing a role in how CAR T cells are being inhibited by tumors. We saw that CAR T cells express VIP, and this expression increases after antigen stimulation. Furthermore, in the presence of VIP, the expression of VIP receptors, VPAC1 and VPAC2, also increase on CAR T cells. Using a NFAT Jerkit expression system, we saw that in the presence of VIP and antigen stimulation, we see a decrease in the amount of GFP expressed by CAR T cells, indicating that VIP is suppressing NFAT-mediated activation of these cells. Finally, when we co-culture CAR T cells and tumor cells and exogenously add in VIP, we can see that VIP inhibits the proliferation of CAR T cells over time. These all indicate that VIP and VIPR are a novel immune checkpoint pathway that are relevant for CAR T cells. In order to target this pathway, we wanted to approach this rather than in combination, but rather than in combination in our armored CAR T cell approach. And so armored CAR T cells are ways that CAR T cells can be utilized to deliver therapeutic proteins to the tumor microenvironment. As I mentioned previously, these can be cytokines that are delivered and engage the endogenous immune system, chemokines, scFvs or nanobodies, or even enzymes. In our approach, we were interested in making a CAR T cell which produced and delivered its own antagonistic peptide receptors. Using in silico design, we were able to engineer antagonistic peptides that compete with VIP to bind VIP receptors. These antagonistic peptides have a greater binding affinity to the VPAC receptors, as shown by the AlphaFold modeling on top. We integrated these peptide drugs into our CAR T cell constructs. For these studies, we're using an anti-MUC16 CD CAR, which recognizes the ectodomain of MUC16, which is expressed on ovarian cancers as well as pancreatic cancers and other solid tumors. We separated the CAR with the P2A element, added our antagonistic peptide, which was fused to a 6X his tag in order to identify the secreted peptide in our experiments. We can see that CAR T cells, T cells modified with the CAR VIPR construct, express the antagonist in the culture supernatant. Furthermore, using immunohistochemistry, we can see that these antagonistic peptides that are secreted by the CAR can turn around and bind to the VPAC receptors, and that's shown in the composite image on the right, where the white indicates where VPAC and the antagonists are co-localizing. 。 Next, we show that these antagonists are actually modulating the VIP-VIPR axis. So on the top, you can see what we believe the predicted effects of VIPR antagonisms are on CAR T cell signaling, where we think it will impact both NFAT and NF-kappa B related pathways in order to diminish activation of immune response genes. As we can see in the RNA-seq heat map, we actually are impacting differentially impacting molecules within these pathways that are involved in the VIPR signaling. Furthermore, when we look by flow cytometry, we can see that our CAR VIPR cells, here indicated in purple compared to the second generation CARs in red, before antigen stimulation, they express less VIP themselves. And this is continued after also antigen stimulation, as well as they have decreased expression of the VPAC receptors. RNA-seq also revealed key differences between CAR and CAR VIPR T cells. The three main pathways that were differentially expressed between our novel CAR VIPR T cells and CAR T cells were related to activation or T cell activation, T cell memory phenotypes, and the T cell metabolism. If we first focus on activation, we can see that CAR VIPR T cells are less activated at baseline, but more activated after antigen stimulation. What this looks like is if we go back to our NFAT jerkit model, where we either express CAR or CAR VIPR, at baseline, the CAR VIPR jerkit, NFAT jerkits express less GFP. However, after antigen stimulation, these cells significantly express more GFP, indicating more NFAT-mediated activation. Next, looking at primary T cells, when we express the CAR VIPR on primary T cells, we can see after antigen stimulation, these cells express more CD69. And in proliferation assays, when we co-culture CAR T cells with tumors that abundantly express VIP, we can see that first, the CAR T cells do proliferate after stimulation, but plateau. However, there's significantly greater proliferation in the presence of VIP by CAR VIPR A T cells. Next, we looked at memory phenotypes. Overall, we could see that CAR VIPR T cells have a greater CD8 to CD4 ratio, and they're enriched for genes related to memory. By flow cytometry, this looks like greater expression of stem cell-like and central memory phenotypes. And when we do a re-stimulation assay where we reintroduce tumor antigen to CAR T cells over time, we can see that the CAR VIPR A cells are capable of expanding greater over time than the second generation CAR T cells. At the end of this assay, at 10 days, we can see that, again, the CAR VIPR A cells are express less triple positive markers of exhaustion, which are LAG3, PD1, and TIMP3, compared to the second generation CAR T cells. Finally, we looked more deeply into the metabolism of these cells. So interestingly, the CAR VIPR T cells are metabolically quiescent at baseline, but become energetic after antigen stimulation. This may be due to when we look at the cells through electron microscopy, we can see that the CAR VIPR A T cells have more mitochondria within the cells, and these mitochondria are structures are more well defined as compared to the CAR itself. And this is quantified on the right, where it's showing that the mitochondrial number in CAR VIPR A T cells is greater. Next, using a seahorse assay, we can see that in red, that's the CAR has after antigen stimulation has decreased baseline respiration and spare respiratory capacity. And so it starts off in an energetic state, but leads to a more glycolytic state after antigen stimulation. Interestingly, our CAR-VIPRA cells in the seahorse assay at baseline have very little metabolic activity. However, this activity increases greatly after CAR T cell antigen stimulation. And so this is uh, shown here where the cells are actually very quiescent at baseline, but become energetic after antigen stimulation. Next, we wanted to look at how these phenotypes perform in an in vivo setting. And so um, primarily, we wanted to see whether they retain their phenotypes in vivo. In order to do this, we engrafted skid-beige mice subcutaneously with pancreatic cancer tumors and treated 23 days later, collected spleen and tumors at 5 days. And we can see in vivo the CAR-VIPRA cells expand significantly greater, both CD4 and CD8, in the mice. And then they also retain this memory phenotype um, in vivo. Next, we looked within these tumors and we could see that these cells are less exhausted and they actually infiltrate more into the tumor than the CAR T cell, the parental CAR T cells. And this is significantly greater, which is quantified on the bottom right. Next, we wanted to see whether these phenotypes are retained in the products of patient derived T cells. And so the experiments that we have done so far in the lab were done with healthy donor T cells, where we manufacture the CAR T cells from these donors. In collaboration with the Chief Clinics, we were able to obtain peripheral blood samples from patients with pancreatic cancer. And we used these samples to manufacture CAR T cells as would be used in a clinical product. Similar to what we saw with our healthy donor CAR T cells, the CAR-VIPRA cells are less activated at baseline, as shown by their smaller size and granularity, as well as higher viability after transduction. These cells at baseline, again, are less activated, however, express greater markers of memory phenotype T cells. Using these T cells, we moved into a patient-derived xenograft model for pancreatic cancer, in which we subcutaneously implanted pancreatic cancer tumor fragments derived from patients into skid-beige mice and treated 24 days later with 5 million CAR T cells. As we can see, the CAR-VIPR T cells significantly have greater control over the tumor, which is also shown by tumor weight. So all of the experiments so far were done with human CAR T cells in xenograft mouse models. We next wanted to look at how these CAR-VIPR T cells are impacting the endogenous immune system. So as the antagonistic peptide is being secreted from CAR T cells, it has the opportunity to not only bind to VPAC receptors on the CAR T cells, but also on T cells that may be found in the tumor microenvironment. In order to do this, we turn to syngeneic mouse modeling using mouse CAR T cells with all mouse signaling domains. In C57 Black 6 mice, we subcutaneously engrafted pancreatic cancer MT5 cell line, and we treated 5 days later with CAR T cells intratumorally so that trafficking wouldn't be an issue in these experiments. And then we looked at, we harvested tumors 3 days later, and we can see within the tumor per gram of tumor, we have more mouse uh, uh, T cells in general uh, in the VIPR-A treated mice. However, there's no significant difference between the amount of CAR T cells within the tumor because we had injected them intratumorally. So these CAR T cells that were adoptively transferred were Thi 1.1 tagged, and so we could distinguish between endogenous and adoptively transferred T cells. And we could see that the increase in mouse CD3 that we were seeing in the tumors was mainly driven by the infiltration of endogenous T cells into these tumors, which was greater in CAR VIPR-A T cell treated mice. Next, we looked at overall survival. So we're again are looking at our MT5 pancreatic cancer mouse pancreatic cancer cell line, which we engrafted sub-Q. We preconditioned the mice 1 day before giving a therapeutic dose of mouse CAR T cells. As you can see, in mice that were treated with CAR or CAR VIPR both had significant improvement in survival and long-term survivor mice um, uh, in both conditions. However, we then took these long-term survivals and gave a second inoculation with parental MT5s, which have less than 10% of our CAR T cell antigen, which is MUC16. And so here we're looking at if there was any antigen spread or antigen or engagement of that endogenous immune system. You can see with a new set of control mice, the tumors grow out. The CAR T cell treated mice the previously treated CAR T cell mice can control their tumors to a small extent, but the CAR VIPR-A T cell mice can control them to a much greater extent, which leads to a better overall survival. So in summary, we've shown that VIPR axis is a novel immune checkpoint pathway for CAR T cells, and that CAR T cells modified to express a VIPR-A antagonist, which we call CAR VIPR-A T cells, have superior phenotype, metabolism, and in vivo efficacy in patient-derived models. We think this is through decreasing VIP-mediated immunosuppression on the T cells and a decreased baseline activation state. This is also through increase in memory phenotype of increase in the subset of memory phenotype, and an increase in metabolic plasticity. All of these eventually lead to greater in vivo expansion of infiltration and persistence and the recruitment of endogenous T cells. I'd like to thank the folks who worked on this project and made it possible. This project was led by a very talented MD PhD student at my lab, Heather Lynn, who recently defended her thesis, as well as Deja Blake, Ruby Freeman, and others. I'd like to thank our collaborators, Dr. Ned Waller, Dr. Lily Yang, and Achieve Clinics, who provided us with our pancreatic cancer patient samples. Furthermore, I'd like to thank our collaborators, including Anupam Pat-Kiri, our shared resources, and our funding sources. Thank you, and please feel free to reach out to me if you have any questions. Thank you.